Firstly, thank SelleckChem for providing a chance to share my research with so many scientists. Here I will introduce my work about HGF-MET as a therapeutic target for EGFR-TKI resistant lung cancer harboring EGFR mutations. I would like to thank support and help from my team, my family, and my friends. And also, I want to special mention that in our study, some compounds such as PI3K inhibitor, GDC0941, irreversible EGFR-TKI, BIBW2992, and the mutant selective EGFR-TKI, WZ4002, were purchased from SelleckChem. Those compounds worked very well. Let's move to the background. It is well known that lung cancer with EGFR activity mutations responds favorably to the EGFR-TKI, Afatinib, and Afatinib. However, although most of EGFR mutant lung cancers initially respond to EGFR inhibitors, these tumors ultimately become resistant almost without exception. Therefore, resistance to EGFR-TKI is a major problem in the management of EGFR mutant lung cancer. This slide shows three major mechanisms of resistance to EGFR-TKI, including T790M secondary mutation, MET gene amplification, and hepatocyte growth factor overexpression. In about 50% acquired resistant lung cancer, T790M secondary mutation reduces binding ability of the inhibitor to EGFR molecule. On the other hand, amplified MET associates with ABL3 and thereby restores PI3K-AKT pathway. More recently, we found that HGF bind to normal MET and send survival signal without involvement of ABL3. Our study was studied from this patient. He has the primary lung cancer and liver metastasis. After treatment with Afatinib, primary tumor shrinked, but liver metastasis enlarged. So we simply thought liver produced a special factor for tumor. HGF is the ligand of c-MET. Since c-MET amplification had been reported to relate with Afatinib resistance, we supposed that liver-produced HGF may play a role for tumor resistance. To investigate the role of HGF, we used two different human lung cancer cell lines, PC9 and HCC827. Both cell lines have deletion in exon 19 of EGFR gene as activity mutation. As you can see, these two cell lines were very sensitive to Afatinib. However, HGF induced resistance to Afatinib in a dose-dependent manner. Next, we performed Western blot to clarify the mechanisms. Reasonably, Afatinib inhibited phosphorylation of EGFR and ABL3, and hence suppressed AKT and ERK. Surprisingly, although HGF induced MET phosphorylation and restored phosphorylation of AKT and ERK, it did not restore the phosphorylation of EGFR or ABL3. Moreover, immunoprecipitation data also shows MET associates with P85 but does not associate with ABL3. To confirm this possibility, we used siRNA. We could downregulate MET and ABL3 bispecific siRNA respectively. When scramble was used as control, Afatinib inhibited growth of cancer cells, but HGF induced resistance. When ABL3 was downregulated, again, Afatinib inhibited growth, but HGF induced resistance. However, when MET was downregulated, HGF did not induce resistance. These results clearly indicate that HGF induces resistance of Afatinib through MET but not ABL3. Let me summarize signaling pathway. Concerning MET amplification-induced resistance, as Dr. Engelman reported, amplified MET is autophosphorylated and transactivated ABL3, thereby activated PI3K-AKT pathway. On the other hand, in case of HGF-induced resistance, HGF binds to normal MET and strongly phosphorylates it. Phosphorylated MET directly activates PI3K-AKT pathway independent of EGFR or OB3. Then we explored where was HGF from. Our immunostaining data using patient tumor specimens suggested us that both tumor cells and tumor-associated fibroblasts can be the source of HGF. In another study, although irreversible EGFR-TKI CL387785 was expected to treat T790M second mutation tumors, and the H1975 cells which harboring L858R mutant and the T790M mutation was highly sensitive to this compound. We found that HGF reduced the susceptibility of these cells to irreversible EGFR-TKI. In a study of percentage of recent resistance mechanisms in EGFR mutant lung cancer in Japanese population, HGF was found in highest percentage of 60% of acquired resistant lung cancers. Here is a schema of this finding. This is lung cancer expressing EGFR activity mutation. When the cells were treated with Afatinib or Afatinib, EGFR signaling will be blocked. If HGF is produced by cancer cells or host fibroblasts, HGF binds to MET and activates PI3K-AKT pathway, and cancer cells become resistant. So far, HGF has been shown to play at least 3 important roles in EGFR-TKI resistance. First, HGF induces resistance to the reversible EGFR-TKI, Afatinib and Afatinib, by restoring MET, GAP1, PI3K-AKT pathways. Second, continuous exposure to HGF accelerates the expansion of pre-existing MET-amplified cancer cells and facilitates MET amplification-mediated resistance. Third, after lung cancer cells acquire resistance to reversible EGFR-TKI, HGF induces the resistance to irreversible EGFR-TKI in cells with T790M secondary mutation. These findings indicate that HGF is an ideal target for overcoming EGFR-TKI resistance. There are several possible strategies for inhibiting HGF-MET signaling, including anti-HGF neutralizing antibody, HGF antagonists such as NK4, MET tyrosine kinase inhibitors, and inhibitors of downstream molecules such as PI3K, AKT, and mTOR. We have established a mixed injection model and shown that fibroblasts induce Afatinib resistance of lung cancer cells in vivo. Using this model, we evaluated the efficacy of HGF neutralizing antibody and NK4 in overcoming HGF-induced Afatinib resistance. From the left panel of this picture, we could see that although PC9 alone tumors were markedly shrinked after Afatinib treatment, the co-injected tumors maintained their size. When we treat the co-injected tumors with Afatinib plus HGF antibody or NK4, the resistance was completely reversed. Next, we further examined the effectiveness of the PI3K inhibitors. As reported in this paper, we showed that PI3K inhibitor PI103 and GDC0941 combined with Afatinib overcame fibroblast-induced resistance. Finally, we investigated the possibility of MET-TKI overcoming HGF-related Afatinib resistance. We assayed whether a new MET kinase inhibitor, E7050, which is currently being evaluated in clinical trials, could overcome the 3 mechanisms of resistance to EGFR-TKI. As you can see, E7050 effectively reverses resistance to EGFR-TKI induced by exogenous HGF. In a co-culture system, fibroblasts induced gefitinib resistance, and E7050 reverses this resistance significantly. From the Western blot data, we can see that both exogenous HGF and HGF transfection restored phosphorylation of AKT and ERK, but E7050 overcame EGFR-TKI resistance by MET-GAP1 inhibition. Downregulation of MET or GAP1, but not UBP3, restored the sensitivity to gefitinib in HGF transfected lung cancer cells. Furthermore, although lung cancer cells with second mutation were highly sensitive to the irreversible EGFR-TKI, BIBW2992, and mutant selective EGFR-TKI, WZ4002, HGF induces resistance to these two compounds, but E7050 reverses resistance to these compounds in H1975 cells. Since HGF was reported to accelerate the expansion of pre-existing MET-amplified cancer cells, we examined the effects of E7050 on this phenomenon. Although HCC 827 cells did not produce viable clones after 30 days of continuous exposure to gefitinib alone, these cells produced many clones after exposure to both HGF and gefitinib. Importantly, E7050 prevented the emergence of viable clones even under conditions of continuous exposure to gefitinib and HGF. Finally, we evaluated the efficacy of E7050 in vivo using our gefitinib-resistant model. Treatment with E7050 alone did not inhibit tumor growth, but the combination of E7050 and gefitinib induced marked tumor regression. In the xenograft tumors, phosphorylation MET was detected in control tumor and gefitinib-treated tumors, but not in the tumors treated with E7050 monotherapy or combination therapy. Moreover, we observed that AKT phosphorylation was markedly inhibited by combination therapy. In conclusion, HGF plays an important role on resistance to reversible and irreversible EGFR-TKI in lung cancer with EGFR mutations. HGF-MET may be considerable therapeutic target for more successful treatment with EGFR-TKIs. Thanks for your attention.